On behalf of the Society of Indian Academy of Medical Genetics, this is one of the several talks that we have planned to educate the clinicians all around the country and outside regarding several topics of interest for the clinical geneticists. I'll be discussing next generation sequencing over next 30 minutes in today's session. Learning objective for this session is to understand the application of next generation sequencing based tests in the clinic. So I'll be focusing only from the clinician's point of view how an NGS or next generation sequencing based test can be used in the clinic. I am very sure everyone has heard about next generation sequencing, exome sequencing, genome sequencing. This is a brief overview of the technique and its use, its limitations, and the challenges in the clinical practice. So, in today's talk, we'll be covering principles of next generation sequencing, clinical exome sequencing. You can see the inverted commas here, whole exome sequencing, what is a multi-gene panel test, and what is whole genome sequencing in my subsequent slides. I just would like to inform the audience there are two articles written by Dhanya and me in the Indian Pediatrics. This is specifically written for clinicians and pediatricians. One is on genomic testing for the diagnosis of genetic disorders in children, chromosomal microarray, and next generation sequencing. We just try to write it in a way so that uh, any clinician should be able to understand this, and definitely for the pediatrician. The second in the series is understanding exome sequencing tips for the pediatrician, where we have questions like is Sanger validation necessary? When do you when is segregation analysis important? Is triexome more beneficial? So such common questions are answered in this article. So I suggest all of you refer if you want more information on next generation sequencing and its application in the clinic. So this is the slide which my colleague Danya has taken efforts to draw this very beautifully for all of you. You can see here, this is double-stranded DNA on the top, and I'll just use my pointer. This is double-stranded DNA, or what we refer to as genome. You can see gene A and gene B written in two different colors for their exons. Light blue for gene A exons, dark blue for exons of gene B, and intronic areas are colored differently, and we have red colored intergenic non-coding region. This is just organization of the whole genome. What we do in next generation sequencing is we fragment this entire genomic DNA into several pieces which contain exons as well as introns and non-intergenic regions. If you sequence this by the new technology, that is next generation sequencing technology, directly, that is whole genome sequencing. Okay, you take the fragments and sequence all of them several times. I will give you the details in my next slide. But if you capture all exons and exon intron boundaries, then it is called exome sequencing. You sequence gene A and gene B and C, D, and so on. But all genes are sequenced, all the protein coding genes are sequenced, then it is exome sequencing. If you capture only coding region of gene of interest, let us say you have B, then you sequence only the dark blue colored exons, then it is called gene panel. Gene panels can contain more than one gene of interest, but each gene is sequenced very well in gene panel, but only few selected genes relevant to the phenotype are sequenced. So this is the overview of next generation sequencing and three common applications. Probably the most widely used application is exome sequencing, where we sequence almost all the 19,300 genes in protein coding genes in our body, and gene panel is only selected genes of relevance to the particular clinical condition or sequence. Whole genome sequencing is means you sequence everything, both protein coding genes and non-protein coding areas of the genome are sequenced. So this is just to give you an idea of what is whole genome sequencing, exome sequencing, and gene panel. Once we sequence there are several methods. Each company has a different way of sequencing. But what we need to know is we get something called reads. Regular sequencing rates are around 150 base pairs. You have paired end sequencing. That means you sequence from either side, and then you get several reads. You get sequence them 50 times, 100 times. I will show you the detail. After this region or target region is sequenced, you align them against the reference sequence. You can see here, this is the reference sequence, and you can take the next read and see where it aligns well. Second read, third read, fourth read. Finally, you get a very nice coverage of the target, the patient's exome or patient's genome, and then you can read what is the patient sequence. Okay, so this is called mapping and alignment. Mapping is finding out the location where it fits, and then we align all the reads against the reference sequence. And then what we do next is we find out the variation from the normal. Again, if you compare the reads against the reference sequence which are available, you get different variants. So that is called variant calling. Okay, then what you get is a variant called file, VCF file, okay, which has all the variants. So if you do exome sequencing, we get nearly 50,000 variants. Okay, then out of 50,000 variants, you need to filter or prioritize one variant in a gene which is responsible for the disease. This is done based on inheritance model, allele frequency, variant predictors, and then we narrow down to candidate variants. What I mean by inheritance model is if it is autosomal recessive, we look for biallelic mutations. Allelic frequency means normal individuals should not have the disease causing variants in the normal population. It should be very rare because these are very rare diseases. Variant predictors means there are several bioinformatic tools to predict whether a variant can cause disease. Then we narrow down to the candidate variant, and then we see the gene disease association, and we assess the phenotype, and then finally we arrive at one or two variants which are probably the cause for the disease in the particular patient. Then we arrive at a diagnosis. Now I would like to show you what do we mean by coverage and depth. So what we usually define is something called a target region or bed file. What are the genes you want to cover? So if it is gene panel, it may be 5, 10, 20 genes. If it is exome, it is all the 19,300 protein coding regions, so protein coding genes. Mostly exons and very small portion of the introns adjoining the intron-exon boundaries. So we do cover small areas of introns which are responsible for normal splicing. Okay, so that is the coverage or target region. Technically, it may be called a bed file. Bed file is like region of interest. Then we have depth, how many times a region is covered. So you can see here, this is a chromosome, and you can see each region how well it is covered. You can see the taller peak, that means it is covered in more depth. Okay, I will show you in my next slide. For example, here it is 64 reads representing the particular exon. You can see these blue boxes are exons. Then you have 207 times, 129 times, 380x. That means 308 reads are there for these two exons, which are covered in one read. You can see two exons which are very close can be covered in one read. Three exons can be covered in one read. Okay, 411x. This is the meaning of depth of coverage or 50x, 100x. Usually we need somewhere around 100x coverage, which means on an average there is 100x coverage, but some regions may not be covered, some regions may be covered more than 200x or 300x, but average is 100. Any good coverage of a particular region should have at least 30 or 20 reads. It depends on how we define the minimum coverage. Okay, I will just show you an example of a 5 alpha reductase deficiency where we see the exon 6, 4, 3, 2, and these are controls. This is one control, second control, third control. You can see everything is covered 70x, 85x, 80x, and the patient's uh, gene is not covered at all in exon 2, suggesting a possible deletion of region. So this is poor coverage showing the deletion. Now, again, to illustrate what is coverage and depth, coverage is it has two aspects. One is breadth, second one is depth. Breadth is what region is covered breadth wise. Depth is number of times it is uh, sequenced. So you can see here exon 1 has 200 and uh, 433 reads, out of which A is 215, C is 1, G is 217. So this is the meaning of depth of coverage. This is covered 433x, and we have a heterozygous variant here. Homozygous variants look uniformly same color and it stands out differently when we see this is called the integrated snap genomic viewer, EIGV, or a snapshot of integrated genomic viewer browser. And on the left side you can see heterozygous variation where you see two different alleles, one normal and one abnormal, and here you see homozygous change. This is how it looks in integrated genomic viewer software. We need to remember if there is poor coverage, then you can see the read qualities are very poor. You can see this light colored uh, coverage bars suggesting this is mapping quality is very poor. Usually this happens in when we have presence of pseudogene like Gaucher disease or spinal muscular atrophy. I will now introduce you few terms. Whole exome. Okay, whole exome is means that all 19,300 protein coding genes are covered. We need to understand now there are non-protein coding genes or RNA coding genes. So we should be very careful. Our current whole exome sequencing technology covers approximately 19,300 protein coding genes. If you see exome is nothing but all the coding regions of the genes. Okay, some people say there is no additional meaning for adding the term whole exome because exome is whole. There is nothing called small exome or incomplete exome. Exome is nothing but whole exome. So some people use the term exome sequencing. Some people prefer the term whole exome sequencing, but they mean the same. Now we have some usage like clinical exome where some people use it for exome sequencing, that is whole exome sequencing done on clinical basis, that is clinical exome sequencing. It is done on clinical basis. Some people use this term for using sequencing only approximately 5,000 genes which are known to be associated with mental disorders, which is of clinical relevance. So some people use it for clinical use of exome, use it for sequencing only 5,000, approximately 5,000 genes. But please understand, a clinical exome sequencing is really confusing terms because there are two types of usages. One is to mean whole exome sequencing done on clinical basis. The other one is means only 5,000, approximately 5,000 genes which are associated with human diseases are covered by this technique. There may be some terms like research exome, that means exome sequencing done on research basis. We also have terminologies like focused exome sequencing, where we again this also means disease causing or disease associated human gene. Mendelium sequencing, targeted exome sequencing, all mean about 5,000 genes. Some people use the term clinical exome sequencing for this. All the human disease causing genes listed in OMIM are sequenced here, which is approximately 5,000. This may be sufficient for your clinical use, but recently discovered disease causing genes, or if you are interested in research, this may not be sufficient. When do we do exome sequencing? This is one important question. Now, everyone uses exome sequencing for every suspected genetic disorder. So, I'll just briefly list the indications for exome sequencing. Okay? Any suspected Mendelian disease is an indication for exome sequencing. However, single gene candidates are eliminated. So, if it is beta thalassemia, you probably sequence the HBB gene and not whole exome sequencing. If it is achondroplasia, you sequence the just region of interest, one exon of FGFR3, because that is where 98% of the mutations lie. So you can just do one targeted mutation analysis test by Sanger sequencing. Or whenever the phenotypes are known to be genetically heterogeneous and multi-gene panel testing is very expensive. Okay. Or any disease you think it is genetic or probably genetic, then it helps in ending the diagnostic odyssey where you establish a diagnosis and you do not have any clue for diagnosis. That is another place where you can do exome sequencing. So, from some examples in the clinic, if you are a pediatrician, it may be spastic paraplegia in children, leukodystrophy in children, epilepsy, muscular dystrophy. Please remember MLPA or multiplex ligation dependent probe amplification for DMD is negative, then only you need to do next generation sequencing or exome sequencing. And congenital myopathy or floppy baby, where you have already ruled out SMA because SMA is a great mimicker of all muscle and nerve diseases, then you can do exome sequencing. Skeletal dysplasia is one more common indication for exome sequencing, but you have excluded common diseases like achondroplasia or hypochondroplasia, or a metabolic acidosis where you do not have a diagnosis, which can be caused by several tens or hundreds of genetic conditions, then you can order exome sequencing. I'll just illustrate one screenshot or an example from OMIM, like developmental and epileptic encephalopathy in children. You have 98 entries. Please note, 98 entries means approximately 98 genes are or mutations in 98 genes are responsible for epileptic encephalopathy in children. So instead of sequencing 98 genes, you can do one test, that is exome sequencing, or even a panel test which is available. This is where you have to, where exome sequencing is really helpful. It may be spastic paraplegia again. You can see 73 entities are there, or entries are there, or genes are there, where we get diagnosed by exome sequencing. It may be common conditions like deafness, retinitis pigmentosa, where genetic heterogeneity is too much. That means more than one gene is responsible for the disease. Okay, so that is where we can we can get help of exome sequencing. Once you do exome sequencing, what do you expect? It is very important. We tell these things to the patients what is expected from the exome sequencing before they undergo this test. There are only three outcomes for practical purpose. One is a potential disease causing variant is identified. That means we have a diagnosis. The second one is no disease causing mutation is found. That means we do not have a diagnosis. We need to do further tests. Third one is incidental findings. Incidental findings are or secondary findings are list of certain cancer, familial cancer syndrome or familial cardiac conduction defects or diseases like Wilson's disease, where a medical intervention can improve the outcome of the condition. So the indication may be leukodystrophy or skeletal dysplasia, but there may be BRCA1 mutation, which suggests there is increased risk for developing breast cancer. These things may be returned to the patient. American College of Medical Genetics and Genomics recommends these are returned to the patients with the appropriate counseling. But there are opposing views as well, because it puts a lot of burden on the hospital counselors and the clinician and even the families. So this is another outcome. It depends on the institution's policy or the country's policy whether incidental findings are returned or not. So these are the three potential outcomes, and we need to be very realistic. Exome sequencing will not diagnose all the genetic diseases because there are several limitations. This is something which we need to keep in mind. One, there may be sequencing difficulties. There is a pseudogene which has lot of sequence similarities with the correct gene or the functional gene, and then there is problem in mapping and alignment. Repeat regions may not be sequenced well, especially GC rich regions or triplet repeat regions. So these are the inherent difficulties in sequencing even using NGS. Second thing is inversions, translocations cannot be detected by exome sequencing, but they can be detected by whole genome sequencing. That is something which you need to remember. Traditionally, copy number variations are not detected by NGS or exome sequencing. Okay? Whole genome sequencing can detect copy number variation. And even now, pipelines or strategies are evolving where we can probably guess very well the copy number changes in the exonic regions using even exome sequencing. Currently, most of the labs would offer you copy number variation analysis from exome sequencing or next generation sequencing data as well. So triplet repeat disorders like Huntington's disease, Fragile X syndrome, these are not detected by exome sequencing or next generation sequencing. Mosaicisms can be ignored or filtered out as artifacts, but if you are looking for mosaic conditions like megalencephaly capillary malformation syndrome, then you can request the lab that we are specifically looking for mosaicism. Please sequence at a higher depth, and we want to look uh, we want you to look at even mosaic variation, then they might be able to help you. Non-coding regions are by design not covered, but there may be some specialized capture kits or even multi-gene panel testing where deep intronic and promote regions may be covered. But in general, exome sequencing does not cover this, but this can be covered in whole genome sequencing. Imprinting defects, which are epigenetic changes again, are not covered by any of the next generation sequencing tests that we are discussing today. So, if you do not get a diagnosis, what does it mean? One, it may be you selected a wrong test. It may be Angelman syndrome or Prader-Willi syndrome, which are due to imprinting errors, which may not be detected by exome sequencing or next-generation sequencing. It may be in the intronic area or promoter area where exome sequencing does not cover this area or it is not in your target region. Difficult to amplify regions, especially the GC rich regions. Okay, triplet repeat disorders like Fragile X syndrome, microdeletion syndromes like Williams syndrome, Diger syndrome. This can be guessed if you do a good copy number analysis, but it is not a standard test to order when you are thinking of this. So structural variation, most of what I am discussing is on exome sequencing because that is most widely used. Chromosomal translocations or deletions, inversions will not be picked up. And if it is congenital rubella syndrome, congenital toxoplasmosis like TORS syndrome, it will not be detected. You might have thought of uh, a pseudoTORS syndrome, but it may be real infection with toxoplasmosis. If it is teratogenic fetal alcohol syndrome, then it again exome sequencing will not detect these changes. If it is a non-genetic disease, remember several developmental problems, birth defects are not genetic in origin, or there may not be any genetic change. If it is not a genetic disease, then you might, or if it is infection, then you will definitely not get a diagnosis. So these are the possibilities which you need to keep in mind when you get a non-diagnostic test. These limitations assume that you have provided complete clinical information to the lab and lab has performed a quality test taking care of good sequencing, quality and coverage. Few questions I would like to address regarding trio exome sequencing. Should I order a trio or singleton? Please remember, most of the times we order singleton exome because of the cost. Otherwise, trio exome sequencing is always superior and patient can afford. That is the test of choice because segregation in the father, mother, and child are completed, whether it is biallelic origin in autosomal recessive disorder, whether it is a de novo mutation in an autosomal dominant disease, which is these are sorted out in one go if you do a triangle sequence. Patient does not have to visit you again because you are going to ask segregation or you are going to ask triazome sequencing the second time. It saves a lot of time. So this is something which you need to keep in mind. It is very easy to analyze for the lab. So if I am running the lab, which I do, then I would prefer triazome sequencing whenever possible, because your variant prioritization becomes very easy. You get lesser number of candidate variants, and you get lesser number of variants of unknown significance, which I'll be discussing further. Triexome sequencing has approximately 10% higher yield. Okay, please remember cost of additional visit, segregation, Sanger sequencing, and coming back again for triexome sequencing and other logistic issues before ordering singleton exome sequencing. I'll This is one slide about gene panel. We have already discussed a gene panel is one NGS-based test where we amplify target targeted genes or targeted amplification of selected genes which are selected on the basis of a phenotype. It may be epilepsy gene panel, muscular dystrophy gene panel, and so on. Panels have a very high depth, it may be 300x or 600x or more than that. And panels should analyze deletions and duplications either by MLPA or by CNV analysis of the NGS data. So it should be able to detect deletions and duplication of exons or exonic regions. Gene panels are likely to be updated for recent discovered genes. Okay, less likely to be updated. What I mean here is, if a gene panel is discovered last year or designed last year, the genes which are discovered this year may not be there in the panel. So latest genes will not be there in the panel which was designed a couple of years ago. It is not usually helpful in a research or gene discovery where you want to identify new disease possibilities. And it has limited value for reanalysis because you probably have done a good job of the target region, but interpretation may be changing. Please beware of the virtual panel where people or the labs do exome sequencing or Mendelium sequencing, and then they analyze only few selected genes of your interest. This is called virtual panel. Major drawbacks are the virtual panels may not have good coverage for the genes of your interest. Okay, and there may not be filling of those gaps by Sanger sequencing. So we need to check with the lab whether they really have a multi-gene panel or they do a virtual panel before you order a panel test. So results of NGS are interpreted in a few different ways. One is it is very important here to note American College of Medical Genetics and Genomics and American Association of Molecular Pathologists have published standards and guidelines for interpretation of variants. We have somewhere strong and conclusive evidence against pathogenicity, where we call it benign, and strong and conclusive evidence of pathogenicity. 99% is a chance that variant is disease causing, then we call it pathogenic. Then we have likely benign, likely pathogenic. Again, strong but limited evidence of pathogenicity or against pathogenicity, which approximately 90% is a constant when we call it likely benign, which means it doesn't cause disease. Likely pathogenic, that means it is likely to cause the disease. In between is a large chunk, which is limited or uncertain evidence regarding pathogenicity or variant of unknown significance. I am very sure it is frustrating for every lab and the clinician and even for the patient to get reports of VUS, but what are they? Where there is no enough evidence for pathogenicity of the condition. So, how assessing the variants of unknown significance? So, we need to, how do we do this? We need to compile and review the existing evidence. Which evidence favors pathogenicity? Which evidence is against pathogenicity? What is the strength of evidence of the pathogenicity or the strength of evidence against the pathogenicity? ACMG guidelines give a very clear flow. How do we score each evidence for and against, and how do you derive a particular score to call it VUS? So it is cumulative evidence from other cases having the same VUS and phenotype. You can look into databases like LVOD, uh, LOVD, or ClinVar, where all the variants are reported along with the phenotype and evidences for and against, and you can make your own assessment. And segregation of the variant through targeted testing of family members really helps. If it is de novo, it increases the chance that it is a pathogenic variant. And if it is biallelic, again, that's an important criteria for making this upgrading this variant to pathogenicity. Function studies, again, we have to read carefully the literature, how well the function studies are done, how well the results are interpreted, and are they correct. That will help us to classify the variant. Revisiting the evidence after a period of time, the variant may be reclassified due to new evidence as benign or pathogenic. So that is one more step which we can do whenever we have variant confusion. Please remember, it is just a definition or category and not, it does not explain the biology. It doesn't explain whether it is disease causing or not. It is just is a category. Okay, it doesn't explain the biology. So please, whenever we have a classification, we need to understand this is based on some scoring system and not based on the biology. For a clinician, what does it mean? So for a clinician, it means if you get a VUS, probably see the phenotype and order the more test to confirm or rule out the diagnosis. For example, you get a variant in aryl sulfatase A, which is VUS, then you do enzyme assay. You take MRI, whether there is any metachromatic leukodystrophy. That is the diagnosis. If it is aryl sulfatase A deficiency, that is how you need to review the phenotype and conduct more tests. Review the literature extensively because there may be some papers, some databases where this variant is reported. Do your own assessment of the phenotype, but be careful when you draw conclusions based on your own assessment because you may not be correct. Review the literature and phenotype again at a later date, maybe after 6 to 12 months. You can request reanalysis by the lab after 6 to 12 months. It should not be used in clinical decision making. If you have a VUS, it is not recommended to do prenatal testing based on a report of VUS in the previous time. But if it is like biotinidase deficiency where you have a VUS, you can give biotin because it is harmless. So you can try therapeutic actions, right, which are harmless, but you cannot do prenatal diagnosis if it is VUS. Additional monitoring of the patient for the disorder in question may be very prudent because you are not very sure. Lastly, few words about whole genome sequencing. Whole genome sequencing is an unbiased way of sequencing the entire genome where you do not have a step of capturing. So it reduces the bias and errors. It sequences the entire genome. It sequences the genes and non-genic areas. Usually lower coverage is sufficient, but only problem is you get huge volume of data, which is very difficult to analyze. Even though we do whole genome sequencing, we often analyze only exome or exon intron boundaries and very few areas of our interest. We can get data on or information on copy number variations as well as chromosomal structural abnormalities from whole genome sequence. So with this, I will end my presentation today. Thank you.